Hello boys, welcome back to another session of online biology video tutorials. This session is meant for class 11th biology students and this is for 22nd July 2021. As we have completed chapter number 3, Plant Kingdom, and understood various concepts related to plant kingdom, this time around we are moving ahead with chapter number 4, Animal Kingdom. In this chapter we are going to discuss about the basis for classification of animal kingdom. What are the different phyla that will be present under animal kingdom, and when we are placing particular animal under a particular phylum, we have to reason out why that animal is placed under that phylum. So starting with that, let us see basically what is an animal. Animals are multicellular, means many cells will be there, and heterotrophic organism. There we said autotrophic in plants because they prepare their own food with the help of chlorophyll through photosynthesis. Here hetero means different, trophic organisms. They depend on different organisms for their food, and they don't have cell wall. The animals, all of us, will not have cell wall. We have cell membrane or plasma membrane, and we don't have chlorophyll. So basically animals are multicellular heterotrophic organisms without cell wall and chlorophyll. So that should be the satisfactory definition of an animal. And 11 major phyla are there under kingdom Animalia. They are Porifera, second phylum Cnidaria, or previously it was called Coelenterata, Ctenophora, third phylum, Platyhelminthes, fourth one, Aschelminthes, fifth one, Annelida, sixth one, Arthropoda, seventh one, Mollusca, eighth one, Echinodermata, ninth one, Hemichordata, 10th one, and finally phylum Chordata. So there are 11 major phyla under kingdom Animalia, and you are seeing the photos of at least one animal for each phylum. So you can remember at least one animal for each phylum. And what is the basis? Why we have kept 11 major phyla under animal kingdom? So the basis for classification is like this. 1. Levels of organization. Okay. When we say levels of organization, animals show different levels of organization. As you know, cellular level like sponges, tissue level like coelentrates, organ system like in our case, like we have got digestive system, respiratory system, circulatory system, like that. So we have got different levels of organization. Then we come to body symmetry. When you talk of body symmetry, it is the parts of different regions and the direction of the body. This is important, direction or axis. So it is of three types. Asymmetrical. Some animals body cannot be divided into two equal halves by any plane. Like most of the sponges, if you take, you try to cut a sponge in any angle, in any direction, you cannot get two equal halves. Then we say such animals are asymmetrical. Then we have got radial symmetry, where body can be divided into equal halves by any plane from top to bottom. Example, you can take some of the sponges, you can take cycon, you can take that round one, radial symmetry. Then we have got bilateral symmetry. A single median longitudinal plane can divide the body into two halves, like all the vertebrates and invertebrates. When you cut up an animal in one direction from top to bottom, you get two exact halves. Then we say bilateral symmetry. Two, bi means two lateral symmetries. And then we come to the germinal layers or embryonic layers. So animals in which the cells are arranged in two embryonic layers. As all of us know, outside one is the ectoderm, inside one is the endoderm. Ecto outside, endo inside. So we have got ectoderm and endoderm. And these animals that are having these two layers during their embryonic phase, they are called diploblastic animals. Di means two. Diploblastic means layer. Animals that are having two layers. What are those two layers? Ectoderm and endoderm. So example, you can write Coelenterata. And an undifferentiated layer called mesoderm or mesoglia is present in between ectoderm and endoderm. So the embryos of all other animals from phylum Platyhelminthes to phylum Chordata have three germinal layers: ectoderm outside, mesoderm in the center, endoderm inside. These are called triploblastic animals. So when we talk of the germinal or embryonic layers, we are classifying the animals into two types based on the number of layers that are present during embryonic stage. One is the diploblastic and another is triploblastic. And then comes the body cavity or coelom. So a body cavity means an internal space present inside the body. On the basis of body cavity, animals are of three types. One, there will be a full-fledged body cavity lined by mesoderm. Such animals are called coelomates, means animals having coelom, body cavity, true coelom, like annelids, molluscans, arthropods, okay, hemichordates, and of course ours as chordates. And then there are animals that seems to be having a body cavity, but it is not lined by mesoderm. Instead, mesoderm is present in scattered pouches in between ectoderm and endoderm. Then we say pseudocoelomates. Pseudo means false coelom, pseudocoelom. Then of course we have got animals where there is no coelom at all. We call it acoelomates. So based on coelom, we have got three types of animals: true coelomates, pseudocoelomates, and acoelomates. Okay, so acoelomates, for example, Platyhelminthes, it doesn't have coelom. Then comes to segmentation. Segmentation or metamorphosis, we call it. So in metameric segmentation, that is true segmentation, the body is divided into number of sections where external divisions correspond to internal divisions. Example, annelida like earthworm, arthropoda, cockroach, and chordata. In pseudometamerism, that is false segmentation, the body is segmented only externally. There is no internal division, like in tapeworm. Okay, you have seen tapeworm. The divisions or the septa are only external but not internal. And then comes the last one, that is presence or absence of notochord. So the notochord presence is mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates like us, and animals without notochord, they are called non-chordates like porifera to echinodermata animals. So presence or absence of notochord will decide where to place the animals. So we go to the levels of organization. As we said, based on organization of cells, animals are grouped into four levels. One, cellular level of organization, animals that have only cells. Then tissue level organization, animals that have tissue, means there is evolutionary tendency. And third, organ level organization. So here from tissues organs are formed. And finally, organ system level of organization. So you can see cells are arranged as loose cell aggregates like in porifera. Example, tissue level, cells are arranged in a particular fashion in a particular group into tissues, cnidarians. Okay, and organs level, tissues are arranged into organ. Example, Platyhelminthes to chordate, all higher animals. Organ system, including human being, they have got a digestive system, respiratory system, like this. So various systems are there to perform a particular function. And organ systems of different animals show complexities. Like digestive system is of two types: incomplete, It has only a single opening that acts as mouth, like this in Hydra, and anus. And example, we can say Cnidaria and Platyhelminthes. Second, complete digestive. It has two openings, mouth and anus. So we have got the anterior mouth and posterior anus. And circulatory system also two types, open circulatory and closed circulatory. So open circulatory, the blood flows freely. It is like a swimming pool. There are no arteries and veins. There are no blood vessels. Whereas in closed, the circulatory system, the blood flows through arteries and veins. It is blood does not flow other than that. It is the symmetry. When we talk of the symmetry, it is the axis, arrangement of similar body parts on two sides of main axis of the body. So body can be divided into two similar parts based on symmetry. Animals are of three types, as we said. Asymmetric. When we cut upon the animal, in any direction we don't get two equal halves. Radial symmetry, a round one, in any direction we get it. And bilateral symmetry, like you are seeing a human being, from top to bottom when we cut, we get two exact halves. So part 1, part 2. Then we say bilateral symmetry. So as we said, asymmetry, body cannot be divided into two equal halves even when you cut in any direction. So most of the porphyrians and snails. Radial symmetry, here body can be divided into two equal halves by any vertical plane along central axis, oral and aboral axis of the body. Examples on porphyrians, cnidarians, ctenophores and echinoderms. And bilateral symmetry, of course, here body can be divided into right and left halves in only one plane, that is top to bottom. Platyhelminthes to chordata. And germinal layers, as I said, if there is one layer, it is mono, two layers, diplo, three layers, triplo. So there are layers of embryo from which all the body organs are formed. Based on the number of germ layers, that is the coverings during embryonic stage, animals are of two types, diploplastic animals and triploplastic animals. You can see in triploplastic animals, outside ectoderm is there, inside endoderm is there, in between mesoglia or mesoderm is there. So such animals are called triploplastic animals. And here you can see only one, that is two layers. One is the ectoderm, another is the endoderm, two layers only. Then we say diploplastic. And two germ layers, outer ectoderm and inner endoderm. See, outer ectoderm and inner endoderm. In between these layers, an undifferentiated jelly-like layer called mesoglia is present, but that is undifferentiated. Example, cnidaria and ctenophora. Whereas in triploplastic, you have got three layers, ectoderm, mesoderm and endoderm. You can see the presence of mesoderm here. So that should make the animal as triploplastic. Platyhelminthes to chordata. And next we go to the next one, that is body cavity or coelom we call it. It is the cavity lined by mesoderm. So you can see the presence of coelom here. It is seen between body wall and gut wall. So body wall and gut wall, there is a space. That space is coelom. Coelom separates the muscles of the gut and body wall. Based on the nature of coelom, animals are of three types as we discussed: acoelomate, pseudocoelomate and coelomate. So in acoelomate, there is no coelom at all. Space between body wall and digestive cavity is filled with matrix, as you are seeing here. So porifera to platyhelminthes. Whereas in pseudocoelomate, you can see the body cavity is not lined by mesoderm. Mesoderm is scattered with pouches. This is mesoderm, that is with the pouches. So it appears as though there is coelom, but actually coelom is not there. So we are calling it as pseudocoelomates. Example, aschelminthes. And then true coelomate. Here coelom arises from mesoderm, annelida to chordata. Here you can see the section, transfer section. And then we come to metabolism. It is the phenomena in which the body or organs is externally and internally divided into repeated segments. These repeated segments are called metameres. So metamerization, the process. So earthworm and cockroach, arthropoda. And last, final basis, it is the mesodermally derived supporting rod. You can see the presence of notochord here, this rod-like structure from anterior to posterior. And this is during embryonic development takes place. Animals with notochord are called chordates, and those without notochord are called non-chordates. So, at a glance, you can see kingdom Animalia, cellular level, tissue and organ level, cellular level mostly asymmetrical, acoelomates, porifera. Tissue or organ system level either radial or bilateral, and if it is radial, it is still acoelomate, so coelenterate or ctenophora. Once it is bilateral symmetry, we got acoelomates, platyhelminthes, pseudocoelomates, aschelminthes, and coelomates. We got phylum Annelida, Arthropoda, Mollusca, Echinoderms.